Good afternoon. My name is Dr. Rebekah Robinette, and I am an Associate Professor of Microbiology here at National Park College. And today, I'd like to discuss our current public enemy, SARS-CoV-2, that causes COVID-19. This is a beautiful picture of the actual virus attached to one of our host cell receptors. Such a beautiful picture, but a very difficult and ugly story behind it. Before we continue with our talk on the virus, we first have to talk about us. And so here's a fairly typical rendition of one of our human animal cells. There are several features labeled. I just want to focus on a few of those. First, let me point you to our nucleus. The nucleus is where our DNA is secured. It is in a membrane, high security, lockdown, if you will, inside of our cells. And that's where our DNA resides. These little features here, called ribosomes, are structures that make proteins from the instructions that our DNA gives, which I'll go into that more in a little bit. The primary focus that I would like to go to now is the outer boundary of this cell. This is called the plasma membrane or cell membrane. And we're going to take a closer look at that in the next picture. So here's an up-close picture of our cell or plasma membrane. It is a big layer, double layer of fat. There's all types of fats that exist, and the scientific term for these are lipids. So our membrane consists of a very highly organized lipid bilayer, so 2 layers of fat. And the center is high security as well. Does not like water, does not allow things to go through easily. So anything that is going to go through our cell must be able to meld, if you will, with this fat layer, or it has to have a dedicated gateway. Okay. Let's move on. Here is how our cells work under normal circumstances. You see, here's the nucleus. That's where our DNA is. Our cells copy the message of the instructions for all of the proteins in our bodies to an RNA language. Okay. That gets edited inside the nucleus, and we have a messenger RNA molecule. That messenger RNA molecule leaves the nucleus, where the ribosomes turn it into protein. So that's how our cells normally work. DNA has the master information, high security in the nucleus. We end up with a messenger RNA message for every protein that needs to be made in our cells. Okay. Now that we know a little bit about our cells, let's move on to viral particles. This is a very simplistic rendition of 2 basic viral particles. We have one that's solid naked. All that means is it's just a protein shell with either DNA or RNA in it. So viral particles are not cells. They're not living. They are active or inactive. They're very, very simple. Some viral particles, in addition to their protein outer shell and their RNA or DNA that they have inside of them, have an envelope, which is lipid fats, just like our cells have, with a few modifications. SARS-CoV-2 that causes COVID-19 happens to be an enveloped virus. It also has a prominent spike protein, like many viral particles do. Pictured here, I believe everyone's seen this particular photograph of our COVID-19 causing virus. The spike protein that's on its surface is of particular importance. It's that spike protein that it's going to use to attach to our cells It is that spike protein that is the basis of our vaccinations. So let's move on. Every viral particle that can invade our cells must have some form of receptor, in this case the spike protein for SARS-CoV-2. And it can only invade a cell in our body that has a receptor that it can dock on. So that's very important, in particular for our COVID-19. The receptor on our cells that it likes to dock on and use to get into our cells is called ACE2. I'm not going to go into what ACE2 is, but let's suffice it to say that they're widespread without our body, but they're very rich in the lungs, the kidneys, the liver, and a lot of the organs that get severely impacted by severe COVID disease. Okay, so let's recap. The viral spike protein of SARS-CoV-2 has to dock on our cells. If it can't dock, it can't get in. So this is going to be an important point later when we talk about our vaccinations. In this rendition of a basic schematic of how our cells work, this is our cell being featured. Of course, you see these viral particles also being featured. What happens normally in our cells when the viral particles dock, they are brought into the cell and broken up by our cell machinery. And then our immune system works on what's called a major histocompatibility complex system, MHC for short. So to make it simple, every cell in our body has a class 1 MHC marker. And proteins that are inside of our cells, whether they're a virus or from our own proteins that are made, consistently get displayed out on the surface so our immune system can recognize if they belong or not inside of our body. Specialized cells in our immune system have MHC2 molecules. And MHC2 molecules are found on major players in our immune system, including the cells that make antibodies. So when the COVID-19 virus gets in normal infection, it gets chopped up, presented on our MHC molecules for our immune system to build that response. This is going to be important when you talk about our vaccinations, because what vaccinations allow us to do is be vaccinated with just a simple spike protein that our cells make as instructed by the mRNA that's in the Pfizer and the Moderna vaccines. So we can avoid the infection and get the protection. Okay? So we'll talk more about that here. So remember, it's that lovely spike protein on SARS-CoV-2 that docks on our cells. So if we could just get antibodies and protections against that spike protein, it would not be able to invade, as pictured here. We want this to not happen with the real virus, but us to get the protections like it did. So the vaccinations, the messenger RNA vaccinations, are not new technology. Research that underlies this advancement started 30 years ago. And over a decade ago, they started researching uses for the technology. So the technology is not new. The use of it in an emergency situation is new. Okay? So, what they do is they take they make messenger RNA that's instructions for that spike protein. They put it in these lipid nanoparticles. Remember, lipid means fat. And the reason why they have to put it in this lipid nanoparticle is because remember earlier I told you, you can't get into our cells unless you can meld with that lipid that they're made out of. Or you're triggered to be brought in during a normal infection. So they put the messenger RNA in this fatty nanoparticle, and voila, here's our messenger RNA vaccines. Those get injected into your muscle cells in your upper arm. The ribosomes that I pointed out earlier that make proteins then produce the protein for the spike protein. And just like over here, those proteins get chopped up or not, sometimes they're whole, and presented so our immune system can recognize them. In this fashion, we build that lovely antibody response against the spike protein of SARS-CoV-2 without ever having the infection. And that's optimal. Anytime you can be vaccinated against something versus having the disease, that's always the track you want to take. So let's move on and talk about the ingredients. I know a lot of people are terrified of these vaccines because they don't know what's in them. And they're also worried about the technology. As I explained, the technology is not new. It was being used for other viruses. Within 1 month, they had the information they needed to make the vaccines for SARS-CoV-2. So, we have our Pfizer, and over here, we're going to have our Moderna. So let's go through them. Main active ingredient is that messenger RNA for the spike protein. I told you that they have the lipid nanoparticles, the fats. So there's several types of fats, including cholesterol, which you've heard of. And it's awful delicious in the foods, of course. And it's really important to our cells. So there is a good use for cholesterol. And so these lipids are what shelter that messenger RNA into our cells. And then here's our other ingredients, salts and sugar. Commonly used salts in medical applications, food applications, and the sugar used is what you might put in your coffee every morning. Okay? The reason why we have to have the salts and the sugars is to stabilize the vaccination ingredients, because messenger RNA is very, very delicate and it degrades very quickly. So we have to stabilize it and freeze it at -Celsius for the Pfizer to keep it intact long enough to get into your arms. Once the vaccines are in your arms, that messenger RNA and the ingredients are gone relatively quickly. What's left behind are the proteins that got made so that our immune system can mount the response against them. Let's talk about the last messenger RNA vaccination, Moderna. Moderna has mRNA, just like Pfizer. Has a little bit of variation on the lipids. One thing that Moderna has is this big old word right here called PEG. PEG is a common ingredient in Miralax, common laxatives of that class. Some people that are allergic to PEG might be allergic to the vaccine, but you would know if you were allergic to PEG before you got to that stage. Okay? And it's a minor allergy. The other thing that Moderna has in it are some acids, acetic acid. Acetic acid is vinegar, essentially. And who doesn't like a good pickle, right? There's some other acid stabilizers. Again, salt, commonly used in IV fluids. And the same good old sugar that's yummy in your cookies and your coffee. Again, the reason why they have these is to stabilize the messenger RNA. And this one is frozen at a much higher temperature in the -degrees Celsius range, mainly because of the difference in stabilizers. Again, these are all common and are used in many applications. So there's nothing in these that's new to our world, okay, and their uses. So hopefully I've eased your mind a little bit about these vaccinations. It was nice to be able to present this to you. Thank you.